Before we get started, I'm going to ask you to do a few things. Don't be a passive learner. Have paper and pencil handy. Pause the video frequently. Rewind and rewatch. Draw as many pictures as you can, because pictures are your friends. Ask questions. Write them down. And go research the answers. And to really test your understanding, try to teach it to someone else in your own words. If you want, leave questions in the comments below, and I'll answer them as soon as I can. Now in this video, our second in our series on DNA, we're going to look at the structure of DNA. We know that DNA contains information needed to carry out cell activities. It's the genetic material, but what's its structure? DNA, deoxyribonucleic acid. Well, we remember from our earlier videos that all nucleic acids are made of repeating units of nucleotides, and that every nucleotide is composed of a phosphate group, a sugar, and some nitrogen base. And if it's a DNA nucleotide, then that sugar is specifically deoxyribose sugar. Now let's take a closer look at these nitrogenous bases. There are 4 different types of DNA nucleotides, each differing in their nitrogen base only. There's our double-ringed adenine, the purine. We have thymine, which is a single ring. You can see the rings out here. We call the single-ringed nucleotides pyrimidines. We have guanine, another double-ring purine. And finally cytosine, another single-ring pyrimidine. Now one of the things that's tough for students is to remember which one of these is which. In other words, how do I remember that the purines are the double rings and that the pyrimidines are the single rings? Well, my hint is that purine has a U in it, and so does the word double. Therefore, purines are double, and then pyrimidines then by default must be single. And then, in terms of remembering which one are pyrimidines and which ones are purines, the pyrimidines, thymine and cytosine, both have a Y in them, and therefore guanine and adenine must then be purines, because that's what's left over. So that's the little trick that I use. You can do it however you want, but it's good to know which ones are which. Now these nucleotides are just the building blocks for a large DNA molecule, so we need to put these nucleotides together. In DNA, the nucleotides is formed by 2 strands joined together kind of like a ladder. So let's build our own DNA ladder. So I made these a little smaller. I have a bunch of nucleotides. I made them smaller so I can fit more in. And I'm just going to grab a nucleotide, and I'm going to start to make a strand of nucleotides. And I can attach one nucleotide to the next simply by making a bond between the phosphate of one and the sugar of another. This sugar to phosphate bond is how we attach one nucleotide to the other. So then I can grab another and another, and I'll build those bonds. The sugar of one to the phosphate of the next, and the sugar of one to the phosphate of the next. And I can continue, and notice that I'm not really paying attention to the order C, G, T, A, G here. That's just kind of random at the moment. We'll talk about when the order is important. So I'm jumping ahead a little bit here, and I'll put a few more, and then I'm going to jump ahead again. So I went ahead and finished building my strand of nucleotides, and we can see that this is only 1/2 of our DNA ladder, but we have this repeating phosphate sugar, phosphate sugar, phosphate sugar as kind of the uprights of our ladder. Now this is a single strand of nucleotides, and we know that DNA is made up of double strands, so we need to bring in some more nucleotides. So let me bring in some more of these. And remember from our earlier videos that we learned Chargaff's rule of complementary base pairing, that opposite every C in this DNA ladder, opposite every cytosine, will be a guanine. They're complementary base pairs. And opposite every guanine then has to be a cytosine, and opposite this thymine must be an adenine. Those are the complementary base pairs that Chargaff taught us. And as we continue to line up the complements across from each other, A's with T's and C's with G's, we can see that we can go ahead and build the bond from the sugar of one to the phosphate of the next, just like we did on the first strand. So I'll continue with this. Actually, I'm going to pause the video and add the rest of them in and then come back. keep moving these in and building this other strand. And hopefully you can see that while we didn't care about the order going down, C, A, or G, A, C right here, but the across is very important. We have to be very particular about what we put across because we have these complementary pairs. And now we're starting to see our ladder a little more clearly. We can see that as we're going down, we have our uprights of our ladder being alternating phosphate, sugar, phosphate, sugar, phosphate, sugar. And so we have our uprights of our ladder, and we can start to see the rungs of our ladder. We'll talk about that in just a minute. So we can say that the sides of the uprights are made of alternating phosphate groups of one nucleotide with the sugar of the next. And the rungs of the ladder are made of pairs of complementary bases, A's with T's and C's with G's. Now at this point we have two distinct strands. We have this strand there, and we have this strand over here, and we have to find a way to connect them together. So we need to look at what's holding these bases together, this C with this G and this A with this T. It turns out that these base pairs are held together by hydrogen bonds. We learned about hydrogen bonds when we talked about our properties of water. We talked about them being weak attractions between water molecules, but here we have those hydrogen bonds within a molecule. These weak attractions holding the base pairs together are hydrogen bonds. And it's interesting because we know that hydrogen bonds are not strong bonds, so the molecule that holds the two sides of the DNA ladder together, the two strands, is actually a series of very weak bonds. And we'll see later why that's kind of an important thing. Now there's one more thing we need to notice here. While these two strands here are complementary, they're kind of running parallel, they're actually running anti-parallel. Now what does that mean? Here I have a phosphate as our first part of this side, and here it's a sugar. And if we move down to the bottom, we'll see the opposite, that this strand ends in a sugar and this strand ends in a phosphate group. The nucleotides are kind of flipped over from each other. Now we can look at each of these strands having the phosphate or the sugar open and designate one end as a 5' end and one as a 3' end. The 5' end is the side with the phosphate, and the 3' end is the sugar. And these 5' and 3' refer to the numbered carbon in the sugar that this phosphate is attached to. This is the 5' carbon right here in this corner there, and this would be the 3' carbon here. But the way I remember it is phosphate and 5 both make the F sound. And we should see that it's again anti-parallel, so that opposite every 5' is a 3', and opposite every 3' end is a 5' end. Let's see if we can grab another one of these and bring it down. So opposite of 5' is a 3', and opposite 3' is a 5', and so they got to be opposite from each other. So there we have our DNA ladder. But this is not the real shape of DNA. Instead, the molecule is twisted on itself in a form we call a double helix. In eukaryotic cells, we have multiple strands of DNA molecules organized with proteins into structures called chromosomes. So let's take a closer look at that structure. The DNA, here's our double helix like a thread, is wrapped around spools of protein. These protein spools are called histones. And in this bottom drawing we can get a better understanding of kind of how this is all organized. Here's our on the very small scale, 2 nanometers across our DNA molecule, and we can see that's coiled up and wrapped around these spools of proteins, histones, which are then wrapped around each other, and it's tightly wound until we get to our chromosomes. So we can imagine the massive amount of genetic code of DNA that's composed in one of these chromosomes. Now, we know that DNA is storing an organism's genetic information. It's controlling the production of proteins, and thus kind of controls the biochemistry of an organism, because proteins are the primary components of enzymes. And we know that the DNA is found in the nucleus of the cell in these strands called chromosomes, or in these molecules called chromosomes. And we just talked about the structure of that DNA. And you have this page of notes in your note packet, but I'm not going to teach this now. But just to kind of give us a heads up in terms of what we're going to be doing, this DNA is coding for the production of proteins. And each 3-letter sequence, and by 3-letter sequence we mean as we're going down, so C, G, T, right here would be a 3-letter sequence. This is enough information to code for production of one amino acid. So to build a protein, approximately 200 amino acids long, we would need a code of approximately 600 lines of code, or 600 rungs of ladder. And so that's what we're going to be getting into as we move into protein synthesis. But before we do that, we're going to have to talk about how DNA replicates, and that's the topic of our next video. So come back for that one.